Hello, this is Ana Doblas, and I would like to talk to you about our recent work published in Sensors, and it's about video rate quantitative phase imaging using a digital holographic microscope and a generative adversarial network. This work has been made in a close collaboration with University of Memphis and our colleague Carlos Trujillo from Universidad AFIT in Colombia. So before presenting our work, let me introduce a little about digital holographic microscopy. So this technique is used to image an stain sample, so it's a level 3 technique. The main advantage is we record the 3D information. We have information about the amplitude and the phase. So that means is we have a whole knowledge about the wavefront, how it's distorted. And that gives us advantage to, if even the sample is defocused, we can apply now numerical methods to focus it, which is really critical for particle tracking applications. And we can have all those advantages using a single shot. So we only need one image, and we can record for example, the tracking of a sperm within a volume, so we can know the mobility. So the DHM, it has two parts, the hardware and the software. So the hardware is mostly the camera is gonna record the optical interference between two beams. The reference beam, which is a plane wave, and then the object beam, which is gonna get the light scattered by the microscope objective, by the microscopic sample, and image through an objective lens, an imaging system, which is gonna be an objective lens coupled with a tube lens, because we want to have at the end a plane wave. So the camera records the interference pattern, and that's called the hologram. And the hologram is only composed about 4 terms. But what we care is only this term. So as you can see, the sample information, it's encoded, it's mixed with other terms that we don't want to use. So how are we gonna solve it, or how we solve it right now? All right, so what we have right now is we analyze the Fourier transform of the hologram. So we see what's going on the Fourier domain. And we realize that these 4 terms are kind of like gathered together to only have 3 distinct terms in the Fourier domain. So this is where we have the DC term, the +1 and the -1. And the only term that we care is the +1 term. So the typical process is, all right, so I know that they are located in different planes. So I can apply a mask. So I'm gonna spatial filter only the frequencies of the object that I'm interested. I know that those frequencies are shifted because I have image with a reference plane wave that is tilted, but I know that tilt, so I can put it back again in the Fourier domain. And now going to the space domain, I'm gonna have the amplitude and the phase image. This process, which seems really archaic, it has been developed several automatic methods, but the main problem of those methods are they fail. Even at some moments they seem that they have images that they are well reconstructed. The same algorithms for other set of images, they are failing. And still we are not getting reconstructed phase images at video rate. So this is the 2 drawbacks that we are now addressing with the cGAN network. And our goal is we want to give to the model the recorded hologram, and the model needs to tell us what is the reconstructed phase image. And that needs to be insensitive to any kind of dust or change of the intensity, any change of the experimental conditions. The model needs to be completely insensitive. And we need to do this really, really fast. We want to have live quantitative phase imaging of the samples. Even though our cameras see the holograms, we want to see real-time phase images. So for that project, we built a match sender interferometer, which is the simplest DHM system. And we record our set of holograms. And for each recorded hologram, we apply one of the reconstructed automatic methods for get the phase image. and those are the inputs of our model. So our model difference with other models that they have been proposed. It's a conditional generative adversarial network. And the main difference of the model is we have two steps. We have the generator and we have the discriminator, and they are competing each other. So the generator, it's a UNet model, it's an autoencoder, and the goal is they're gonna create us a face mask. And the discriminator is gonna see the face image created by the generator and the target face image, which is the one given by the automatic. And we need to try to fool the discriminator. Like there is a moment we're gonna assume that the model is trained when the discriminator cannot make any difference between the target face image and the generated face image. That is the step when we're gonna say yes, that is trained, because we are completely the discriminator part. So we did it, we trained it, and those are images. So the first, this is the video. The first is the red blood cells were moving. And you see that the first image was with the automatic method that they were still pumping and having problems. I'm gonna run it again, like red blood cells. They were not always were restricted, they were changing. This is the ones from our model. There is still some failing. We see some blur at the end, but our model, it provides less noise, less face distortion, and it works mostly for all the frames. So I hope that you like our work. Please read our work in Sensor, contact us if you have any information. I'm looking forward to hear that you like our work. Thank you.